Okay, hello everyone. Today I'm going to present the chapter 11. So last chapter we're talking about the FAD and I'm sorry. Last chapter we're talking about another group of chromosomes. That's BV and the fees and the CBCRs. And this chapter, we're talking about another family. And the chromosome they're using is FAD. That's a flavin adenine dinuclease. This one. And the photoreceptor or the proteins there have the domains, including like the BLUF, that's a blue light using FAD, or the cryptochrome CRY and LOV domain, and some proteins molecules that using the LOV domain. And the LOV stand for light, oxygen, and the voltage. And here it shows like a general diagram of the molecular, chemical molecular of the FAD. And here this shows a CRY structure and turning to another side. And we can see that there are alpha domains and the center there's a pocket and another part of these proteins. So for this image is from the last lecture and from the chapter 10, and it's talking about the tetrapyrrose, the phytochrome, and the CBCR. And today we're going to talking about some CRY and the phototrophins that are using the chromosome of the flavin. And that's today our focus on. So for the flavin, FMN or FAD, so this shows the chemical molecular of the riboflavin RF and FMN and FAD. So it has this flavin part and the adenylene dinucleotide part. And this one is a mononucleotide part. So here this image shows clearly. So this is the central the central chromosomes that this pi system will have like sense the light and cause either like protonization or electron transfer or something. It's here. And this part, the blue box part is a riboflavin, and adding phosphate, that's MMN, and adding nucleotide, that's FAD. So for the flavin is like different structure, like actually I mean, it's the same structure, but depending on the location of like the electron or the proton that come to like here, and it were sensed to different wavelengths, and it were emission different light wavelengths. So this is also from the previous lecture, and it shows the dimer of the BLUF. This is PAC. This structure is the PAC, and it has an adenyl cyclase domain in this part, there's a layer, there's the tongue that's inserted into the here, and the linker, and this part is the domain that binding the chromophone FMN. Is so upper this part is the enzyme part, and this part is the light control, that's the light receptor part. So let's move on to see the structure of the cryptochrome, the CRY. As we can see here that the chromophone binding site is here, and there's some beta sheet here, and the chromophone is in the center, and LLV domain is smaller, chromosomes here, and there's some like alpha helix here. So this shows the BLUF domain, a structure like this. There's more like the beta sheet here, and it forms the hydrogen bond with glutamine, right? And this is linked with cysteine. So here is a label of the FAD structure, and that this is number 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, and the center down this 5, and this is a 10. So so this part is very important for the function of this chromosome, and they linked to different like the residue of the amino acid, and it will cause like the structure change of the whole proteins. So first, let's talking about the CRY. So the CRY's chromophone is binding in the center pocket. And this is the large residue, maybe small, maybe pretty large. And the most important part is this 5, this position 5. So this, and it's with electron with negative charge, or add with a proton, or this electrons go move on this the side 1, this nitride part. So all this movement of the electron and will cause like a different wavelengths of its emission or absorption. CRY is a flavoprotein receptor that was first identified to be responsible for photomorphogenesis of plants. So yes, indeed, the CRY is really a photoreceptor and then transmits the light signal into like some morphology change of the plants. And the CRY was also discovered in insects and animals, was identified as a component of the circadian clock, and was also believed to be involved in the magnet reception in birds and insects. Yes, CRY is considered to be like the sense of the magnetic field. And in the later lecture, there's about application of all of this chromosome, but that magnet is completely different. It's nothing to do with the magnetic field. It's just like the one that or some part after modification, it cannot be dimerized with exactly the same, but it will dimerized with another like with another property, like another one is a negative charge and this one's a positive charge. So it's very hard for positive charge to form a dimerides, but it's easier to form a dimeride with a negative charge and with a positive charge. So it's a little like a kind of the magnitude of the ion that this unpolar will have form like attractive with the opposite polar. However, it's just the name. It's not related to the sense of the magnetic field. And the CRY are also similar to those of DNA photolysis, and are classified as CRYPR family. And yes, we're going to discuss about a lot of research and engineering work of this group of chromophore and this photoreceptor, and it were related to the DNA binding, transcription, or gene editing. And most of the scientists they choose this flavin family. So here, talking about so this is the structure that they analyzed, and it was called a crypto dash. So the name is called a cry dash, and it's and let me think. Remind me of the cry the rainbow dash, and the name is pretty cool. And the D dash is coming from like the Drosophila, Homo Arabidopsis, and Cystic. And this um so name this fifth subfamily with the cryptochromosome dash. And here it shows the structure of the CRY. Okay, so the pocket is located in the center of the structure, and the alpha helix will have a long tail, C-terminated tail, that are linked with insert into the pockets of the chromosome. So after absorbing the light, the chromosome the change will interact with this long tail, and then will cause the conformation change of the alpha helix domain. Let's take a look at the phototropism, and the domains is the LOV, that's a light voltage. So for the LOV domain, so this is the LOV domain, so crypto is CRY, and the phytochrome, that's the chromosome we're using is the tetrapedal. That's how to spell that? Like a tetrapedal, right? Tetrapedal, and it's discussed in the chapter 10. So we can see that the phototropism that with the LOV domain is sensed with the blue light, and it will cause like the plant to move towards the light. And the blue light can also activate the CRY, with light will start to like morphology change, start to grow the leaves. And the face are also involved in like the cells, the plant growth and differentiation and morphology change. So all of these chromosomes are a receptor of the light signals. So for the phototropic, and so in the dark, so it forms a folding. That's and after shading with the light, then the conformation of the photo and the LVL2 will cause some conformation change, and it will cause the removal of the phosphate on the NPH3. So the phosphofators function will start working when shading with the blue light. However, in the dark, the kinase will go to this NPH3 domain and will start to phosphorylate this part. So the phototropin is firstly is discovered in this algae. It's like a rod. This green algae is from it's not like the green algae round, and it do not have the flagellarism, and it forms like the shape of a rod. And with fluorescent, it has an autofluorescent. When the shading with fluorescent light, then some parts will show this like it's fluorescent stimulation, so it shows the position of this phototropin, or called orochrome. Then let's take a look at the PAC, the BLUF domain. Actually, the PAC is because it's a adenine cyclase enzymes. So in the previous lecture, I think the chapter 7, we already discussed this one thoroughly. So it's a different structure after shining with the light, and the glutamate and 83 will has like a turning around or like a flip. Then so we can see that this oxygen will flip to here, and this nitric and were flipped back here. So the hydrogen bond will be changed. And so these two parts were separate, and so this change will cause the absorption wavelengths will be changed. So measure the light response is the isomerization of the glutamine side chain during photoaction of the BLUF domain. And this method is the ultrafast infrared spectroscopy, an experiment. The redshift absorption suggests strengthening of the hydrogen bond between amino acid and the C4 and O group. So this is here. So C4 is, let me see, 1, 2, 3, 4, 1, 2, 3, 4 is the C4 and the O group. So here of the FAD in the intermediate state. So this change was majorly like at these areas. So this is like the slide coming from previous lecture, chapter 7. I shows there's many different types of the PAC and then cyclists. Now let's take a look at the application. Since the flavin are universal cofactor in cells, essential for all organisms. And this light independent control can be used as like gene expression, protein degradation, neural differentiation, cell-to-cell interaction, or organelle like moving within the cells. There's many cell molecular functions can be controlled by light based on this flavin response to photoreceptor families. So first, let's take a look at application of the phototropin that's we using the LOV domain. So here is a summary of these their applications like CRY, have application like gene expression, like binding DNA binding, like activate transcription, or protein interaction or localization is majorly is involved in the histidine kinase domain or some GTPS. And LV is a really will be focused and discuss it the many applications for the AAV like gene expression, gene editing, protein interaction, protein degradation, and second message synthesis or hydrolysis. And for BLUF, so this chapter narrates the two examples. But because the central idea of like after light, there's some domain or some subunit were binding together, or because of the light, they're separate. So the general ideas were the same. So today I'm not going to talking about the BLAF, BLUF domain, and we're focused to the LOV domains. So first, and so this is the protein-protein interaction. That's using like the two different domains. The one is the GL, and another part is the FKF, and this yellow one indicates YFP. Yellow fluorescent protein. So after shedding with the blue light, so this part, so there's a conformation change of this GI, and FKF were binding to the GI. So it's the same idea for the gene expression in this, that the GI linked to, so here GI linked to the plasma membrane, and here GI is linked to the transcription factor like the GAL4, and the FKF linked to transcription factor VP16. So after shedding the blue light, so the GI and FKF1 were binding together and cause the VP16 interact with the GAL4 and start the gene expression. So this system is also applied into zebrafish. We can see that it's a zebrafish. So after the shedding the light, the LOV domain will form a dimer and cause these two binding together, and then this can start the gene transcription. So for in the like the mutant like knockout zebrafish, and after turn on the DNA transcription with this optogenetic tool LOV and forms a normal. So it's like a rescue to fall back into a normal morphology of the zebrafish. So it's another. So here, this one is the magnet system that I was talking about previously. And this, so this is still an LV domain, and it can form a dimer. It can form a dimerized after shedding with the blue light. So the authors, they changed a little sequence that the previous is VVD. So change one part, the PMAG change to RR, the NMAG change to DG. So this two becomes like R is a positive charge, and yes it is. R is a positive charge, and the D is the super negative charge. So the RR and the DG after shedding light, there's conformation change, and will cause this two positive charge and negative charge was more easier to be approached to form a dimer. However, the same RR or same DG, because it's it's the same like a charge. So after the blue light shedding and or not forms a dimer. So why? So I think it's quite interesting because that. So in this way, um, we can like a linked like a different parts different parts of this like subunit and the to the either this one or another one of the subunits of the VVD. Then after the blue light, so you can guarantee that that's the different the binding with the different subunit will cause dimerized by this magnet construction because it shows like for example like this one is a positive charge, another one is a negative charge, and it will link it together. So you will exclude out that if these are exactly same like this, and this will form like dimerized with another copy like a clone of this one. So then the heterogeneous binding of the N and C will cannot maybe, so the possibility will be reduced. However, if these two parts is a little different, so you can form a dimer and even more like tighter because there's a different charge of these two different subunits, and it also can guarantee that it's a different part, a different subunit were binding together, being activated by light. So this system is the magnet system, and as what I say before that, already naturally the CRY is a completely different because this flavin is the LOV, and we can tell it's because that it's a quite. Let me check again. I think it's a LA. So. Yes, it's a LV domains. So it can guarantee that there's a different subunit or will come together by the light. So here, this part I think is also pretty cool. So it's like it needs like the two break to for like using some ligand to make these two coming together. Also, you need the light to release the protease cutting site that was being covered by the LOV domains. So after shedding with the light, then this part will be released, and the protease can cut this sequence and release the nuclear translocated and the transcription factor and start the fluorescent, start the targeted gene expressions. And this is a two break because first it require interaction here and the interaction approach interaction and the cutting side here. Well, of course, I think that we can also change a little bit and add these two different parts that is controlled by light. But this system is focused on this protease cleavage because this is a very fast transcription indicator. So it just need to shine in the light 5 minutes and it will start the transcription. The cutting and transcription will go in. So this result is coming from like after 9 hours. So after the binding, then the transcription, then translation, then get the result of fluorescent protein. However, at this light control with 5 minutes will be enough to start the transcription. So it's a quite like the timing is very good for the transcription readout. So this is another part that is for localization of different proteins. So this is like they do a phage display selection. That's to make sure that these two interactions are being triggered by light. So the first they need to binding, then wash off those unbound. So then take all this already bonded, then put in a no exposure in a dark condition, then only keep those with not bound and remove those background unspecified binding that's not that's independent with light. So only blue light trigger the protein bindings and were being selected. So in this way, they can do control that after shining the light, then the protein will be focused on the proteins will be targeting or like binding to the mitochondria or some part of the plasma membrane. So this of course is called light-induced dimer. And this study is a localization and activation of signal proteins. So after shedding light, then focus then start to target to this cell to specific subcellular organelle or structures, or like the plasma membrane or something like that. So this is a light controlled cell localization of specific protein. Later, we're talking about like the light control specific like transportation of organelle. So this is a different is that low. So in the dark state, the LOV2 were binding to sequence. Then after shining with the light, the conformation change, and it will expose this sequence, and there's the other the target proteins were binding to this. So this is like a cover. It's like a shading, like for a specific sequence, and then this cover is the LOV2 and IPAA. So this is LOV2 with the IPAA peptide. So before the light, so this LOV2 will be covered to IPAA, and there was no interactions between these two parts. So after shedding light, and this part is being exposed, and they start to have protein interactions. And for the GAL4AD will be especially approach to the BD and start the gene transcription. So here is a subcellular protein localization. So before this, with the arbitrary protein fusion proteins were floating inside the cytosol. So after shedding with the light, the conformation change will cause the LOVP or have an interconnection with the PDJ domain. So and it shows that a whole construct was being targeted to the plasma membrane. And you can see here, as in dark, no light. If there's a spotlight, and the arbitrary fusion protein will target to this spot, and you can also recover as a global as a shedding like light at all area, and it will all approach to the plasma membrane. And this is how this using the light can control like a specific protein to target into the to approach to the plasma membrane. So is nuclear localization signals. So this car signal and were linked to this nuclearized like linked signals. So after shading with the blue light, and this the nuclear localization signal peptide will be released, and these proteins will be transported to the nucleus. So like before, that's the homogeneous like location of this protein after shedding the light. So and the nuclear localization signal peptide will be released, and it can be recognized and transport the whole protein into the cell nucleus. So this uses a light controlled nuclear localization. So this is another applications. So here this group talking about another approach is that with shedding the light, the nuclear target sequence is not exposed, and after with the light, and this part will be released, and the import like a chaperone or like interact proteins were binding to this sequence and cause the translocation of the whole proteins. So this is a cell-cell interaction. And so it's like cause a cell matrix adhesions. And it's a photo ECM, like a photo switchable extracellular matrix protein. So it's like there's a protein that are being stabilized on some kind of a surface, and this photo ECM, this protein is here. So the orange part is this part is the LV domain, and the upper part is the RGD motif. So this motif, after shining with the light, this motif will be released out and combined with the integrin proteins. This causes infection and cause the cells to like being adhesions and to this surface. So this is cause cell-cell interactions by these systems. That's pretty cool, right? So this part is cause, this is like a kind of like the light control, like kinase, because the membrane bound kinase usually need to form a dimer to 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 start to function. So this is a light controlled tyrosine kinase. So after shedding the light, the LV domains are binding together, and the kinase domains are start to function. So there are different like approach methods, like for this ligand, for this tyrosine kinase domain to binding together, and for the LOV like domain shedding blue light like coming together, and of course there's the same situations can like some proteins they're not on the membrane, they're in the cytosol or in the nucleus. So all of them, since because this part they have a nuclear target localization signal, so they will be inside the nucleus after shedding the light and will binding together and start to have active tyrosine kinase function. So I'm quite very interested with the nuclear opto and FGFR. FGFR is the fibroblast growth factor, and it has a nuclear localization sequence and fluorescent protein and a kinase domain and some linker, and this two cause the dimerites and interaction of the two parts. And so another group like think about how to do the gene acting and controlled by light. So this they using like losing the light that cause this two part is not dimerized, it cause them to separate. So in the dark, so these two are binding together like naturally, and it cannot, and the other like the Cas9, the complex will not form cannot interaction with the DNA. But after shedding light, excuse me, after shedding the light, then these two become separate and they can bind into the DNA and to start the gene editing. So there are different designs. Some cause some like the blue light can cause they form a dimer, and some can separate them. Sorry. Okay. So these light switch proteins based on flavin can also designed to as a light dependent protein degradation. It's like the target protein were linked to this part after shedding the light. The sequence was being exposed, and the exposure of the sequence will cause the whole protein being degraded by protease. So this is they're using like and this we need to know that it's a success for the on the east and because of the east it's not function. Oh, okay, the east and because of the function east because so far, well, take a look at other literatures and that have been cited by this chapter. I didn't show that it's in higher in other organisms. So there's only like a successful on the yeast. So the yeast is being captured on a dish. So the light signal they were using like a photo that some part is cannot penetrate the light. So it causes a result like this. So it made me like quite think of it's like the tattoo, how to make the tattoo. Like some shedding the light and like some of the pigment just degraded and however like like all of the some parts is being covered by like some shedding and some parts can expose to light. So with this then you will cause like a specific pattern on the surface. I think it's pretty cool. So this so in another group they're successful on the HHEK and zebrafish, but the idea is still quite similar to the previous one. And you'll see in the LOV domain and also with binding with a peptide, and this peptide RRG like a sequence like be substituted here, and it's called a digram. And this is an ubiquitin independent manner that will cause this sequence RG will cause the degradation of the whole protein if this sequence is being exposed. And this already been successful on HEK cells or on the zebrafish embryonic embryo. So let's move on to see the crypto chromophone. So here I'm going to write some examples. For example, the gene regulation by CRY. So the center like the construct is a CRY1, C1b1 interaction. So one part AD linked to the C1b1 and the BD linked to CRY2. So after shedding the white, CRY2 binding to C1b1, and this will start to follow gene transcription. So there's also, so here's a new one that's the light controlled CRISPR Cas9 system. At the so the CIBN part is binding to the DS9, and the CRY2 were linked to the VP666 after shedding with the light. the CRY2 will bind into CIBN and will cause the VP64 like have an interaction with the Cas9 and it will start the CRISPR Cas9 system and start the gene transcription. So, so in this paper, in this article, they described several the protein localization method, like the CRY2 with the CIB1, or the LOV domain that's like interconnection with the SSPB. This couple, and also the MMAG and the PMAG that's is also the LOV domain, LOV domain using. So this article is kind of like a summary of different protein localization. You can use different like the different pair of this protein interactions. So for the BL, no, it's not BLU. This is a CRY an application on that. And like for the ligand, it can cause this tyrosine to form a dimer. And if like this substitute with the PHR and cause a dimer, will also start to cause the transcription. Oh no, I'm sorry. This is a kinase signal pathways activation. So after provide the ligand, the BDNF, and with the different lights, so this signal, this ERKM cherry signals will be activated and located into the nucleus. And it's also like shining with the lights. It will also do the same thing that make those ERK and cherry can go localized into the nucleus to start the neurotrophin factors. So forms a dimer. So simply forms a dimer. And this is published in 6 years ago by Chen's group. So I would like to talk about this because it's not only like in HEK cell, but also in the PC12, the neuron cell lines, and also an isolated hippocampus neurons from the mouse. It's a cell culture, but it's isolated neurons that from the mouse, the hippocampus, and it will has the effect of the neurotrophin signal pathways. So I think this is pretty cool. So another is the CRY2 and the CIB1 binding is the light control of organelle distribution in the cells. So the experiment designs that link the CRY2 with the CIB1, and after shining the white light, so the organelles will be binding or be connected to the kinesin and start to move on the microtubule. And so another like a design is the CRY, CBN, and the BRCDN. So in the dark place, there's no interactions. After shining light, it forms like protein complex and start to transport the organelle like approach to the nucleus. So it's also used in like the light controlled protein interaction. But we know that here we already know that it can cause like the neurotropic signal pathway, activate gene editing, gene expression, and also like the cell attachment or organelle like transportation. So this is all required by the protein interaction, and all of this process that can be controlled by light after work with this photoreceptor like machineries. I think they're pretty cool, and because there's only two like examples for the BLUF, so I'm not talking about here. So general ideas are the same. The light will control the protein interaction. So here there's a reference. I try my best to including all of them, but maybe not. So zero citation is the flavin, a universal cofactor in cells. And in addition, actually, I would like to say, Freud, the murder case is being determined, and it's a time for us like to heal, to build, and to love. Especially, I would like to talk to you in this lecture. It's not only because like yesterday the case is being like the jury has make the decision, and also the main words in here is the LOV. So it's time to start to love. Yes. Thank you, everyone. Thanks.